Good morning. This video is going to go over the stages of meiosis. Now, you're going to have to be really careful because it gets confusing because meiosis sounds just like mitosis, right? What we talked about when we went over the cell cycle. So the cell goes through interphase, G1, S, replicates its DNA, G2, and then it goes into mitosis. Prophase, metaphase, anaphase, telophase, and eventually at the end of mitosis we end up with two identical daughter cells. This is how the cell reproduces, right? Meiosis is going to have some similarities, and especially in how we name things and the processes, but the end result is very different. Meiosis is a process where we're going to take 46 chromosomes, each with 2 chromatids, and we're going to divide it into 4 cells with half the number of chromosomes, half the number of chromatids, and those 4 cells then will be potential reproductive cells, so sperm or egg. And the reason that we have to do this is, if you think about it, our normal body cells have 46 chromosomes, right? 2 pairs of 23. If we took one of those cells from a male and a regular body cell from a female, and they combined together during fertilization, we would end up with a cell that had 96 chromosomes in it, right? That's not what a human is. A human beings, Homo sapiens, have 23 chromosomes, 2 pairs of them. So we have to somehow figure out how to divide up the genetic material in half and get them into cells that are going to be used in fertilization, so sperm or egg cells, so that a sperm has 23 chromosomes, an egg has 23 chromosomes, and when they combine during fertilization, we create a new cell that now has the full 46 chromosomes in it. So this is what our goal is. So meiosis, by definition, is the process of dividing the number of chromosomes in half to produce gametes, or egg or sperm cells. And each one of those then will have 23 chromosomes in them. So we're going to start with a body cell, or what we would call a germ cell, kind of a generic cell that would be found in the cells of our body. Now, what we have here is, let's say this organism has 2 chromosomes in it, say chromosome 1, chromosome 2. We have 23, but we're not going to draw 23 chromosomes. So we're going to pretend there's 2 chromosomes. Mom gave you a copy of chromosome 1, mom gave you a copy of chromosome 2. Dad gave you a copy, or sperm gave you a copy of chromosome 1, sperm gave you a copy of chromosome 2, right? Now remember that at some point during the cell cycle, during the S phase, the DNA is going to duplicate itself, right? So we end up with a cell then that we're going to end up with. So the chromosome number 1 that we got from mom is going to end up with 2 chromatids, right? The chromosome number 2 that we got from mom will end up with 2 chromatids. And same thing with dad, right? The DNA is making a copy of itself. So we're going to end up getting, we still have, remember, count the centromeres, 2 chromosomes or 2 pairs. So we got 4 chromosomes, just like we had 4 chromosomes up here, but each one now has 2 chromatids. So here's what's going to happen. During the stages of mitosis, these cells are then going to undergo what is called meiosis, and their meiosis is divided into 2 different parts. During meiosis, the mother cell, which is this, really kind of what we're starting with, is going to divide to produce 4 non-identical daughter cells that are going to have half the number of chromosomes as the mother cell. So remember that this is considered a diploid cell, right? Because it's got 2 copies of each chromosome, so we call that 2N. These 2 chromosomes here are called a homologous pair, right? Because they are both chromosome number 1, so the genes on them are going to be the same. There might be different versions of them, but they would be the same. They're the same chromosome. So chromosome number 2 then would be a homologous pairs, right? So in a homologous pair, you have 1 chromosome that came from mom and 1 chromosome that came from dad, and they're both the same chromosome. So for example, chromosome number 1. Now remember, homologous pairs are not identical, but they're very similar to each other in length and the information that's contained, although there are different versions of it. So what's going to happen is that we're going to start then going through a stage that's called meiosis I. Okay, so everything below this point is going to be meiosis I. Now, during meiosis I, what's going to happen is we are going to start with our parent cell here, or our germ cell, and we're going to end up making two cells, each with one copy of the chromosome in it, and each chromosome will have two chromatids. So we will go from a 2N or a diploid cell to a 1N cell at the end, two 1N cells, and they will not be exactly alike. So we'll start with prophase. What happens during prophase 1, and here's a handy dandy clue. If you see a question or a test question or case study question and it lists these stages, so prophase, metaphase, anaphase, telophase, right? These are the same names that we used in mitosis. However, because there is the Roman numeral on it, so either prophase 1 or prophase 2, then you should know that this is meiosis instead. And the names, we use the same names because the processes that are going to be occurring are very similar, even though the end results are going to be very different. So here's what's going to happen is during prophase 1, what's going to happen is the chromosomes are going to find their homologous pairs and kind of match up with each other. Okay, so like here will be the chromosome number 1, so they're kind of going to migrate towards finding each other, all right? And then chromosome number 2's, the two versions of that are also kind of going to migrate towards each other. Okay, so basically prophase 1 essentially is kind of going to be a pairing up of these different chromosomes. Now, what's going to happen is that there is this system here, and I'm kind of going to draw this over here, that's called crossing over. So what happens is when they start to pair up, got a light bulb right there, these chromosomes are going to actually swap genetic material, right? So we're kind of going to end up with a chromosome that's kind of going to look like this. So we'll put the blue right here and the blue right there, and then we got this chromosome over here now that's kind of going to look like this because it's going to swap this genetic material. Okay, so this is called crossing over, and it happens during prophase 1. This is what gives us genetic variety, right? This is why your brothers and sisters don't look exactly like you. It's like kind of like a swap of information. I'll give you this version of eye color, you give me this version of eye color. The traits are the same and they're in the same location, but this is going to give us a little bit of a variety. So they kind of pair up and stick together, and then this crossing over can occur in order to produce some genetic variety. So once prophase 1 happens, the spindle fibers are going to start to form, right? Remember that happened in prophase and mitosis too, so you can kind of draw those spindle fibers in. Those spindle fibers were formed. And then what's going to happen is we're going to go into metaphase 1, where guess what? They're going to line these homologous pairs up down the middle of the cell, right? So we're going to have chromosome 1 is going to line up right here. Chromosome 2 is kind of going to line up. Metaphase. And it's all going to be lined up according, these spindle fibers are going to form and they're going to align those up. So remember, M for middle, they line up along the middle of the cell. Then, just like in mitosis, we're kind of going to pull these chromosomes then. So here's our spindle fibers still. We're going to pull these to the poles of the cell. So here's this chromosome, here's this chromosome. Here's this chromosome, here's this chromosome. We're pulling them apart. Remember, A for apart. Then for telophase, we're going to reform the nucleus around each of the new groups of chromosomes. So we're going to end up with a nucleus. We're going to end up with a nucleus here. This one's going to have, let's say, this chromosome and this one. So it's got one of 1 and 1 of 2. This one's going to be here. This one will be there, right? This is going to cleave, right? So we could kind of erase here if we wanted to and kind of show this cleaving process that's going to occur. And we're going to end up with 2 cells now, each one with 1 chromosome of each, okay? Now remember, these are going to have genetic variety to them. These are called haploid cells because there's only 1 copy of each chromosome. So we went from a cell that had 2 copies of every chromosome, and we've divided it now into 2 cells that only have 1 copy of each chromosome. 1 copy of chromosome 1, 1 copy of chromosome 2. This whole process is called meiosis I, but we are not finished yet. Now we have to go through one other division process where we're basically going to split apart each of these chromatids so that we will end up with 4 cells, each with a chromatid from the chromosomes. And it's going to work very much like meiosis I, except we are now going to call it meiosis II. So now we're going to go through mitosis II, okay? And mitosis II is going to further divide the cells into 4 cells, each containing 1 chromatid. And this is going to be set up very similarly to how mitosis occurs, all right? So we have, here's what we started with, 2 haploid cells, each with 1 copy of the chromosome. Each chromosome has 2 chromatid to it, okay? Chromosome 1, chromosome 2. So during prophase 2, that nuclear envelope is going to disappear just like it did in the other prophases, right? So we will no longer have a nucleus. We're going to end up with spindle fibers that are going to start to form. That all happens in prophase. Spindle fibers start to form here. They're going to connect to those centromeres of the chromosomes. And then the metaphase 2, the chromosomes are going to line up along the middle, which they kind of already did. And it's going to be driven. That process will be driven then by those spindle fibers, lining them up right along the middle. And the same thing will happen over here to this cell, lining it up along the middle. Spindle fibers are going to move them into the proper place. Then anaphase, right? We're going to pull each of these sister chromatids apart. The spindle fibers will. So we're going to end up with a chromatid over here and a chromatid over here, and then a chromatid on each of those sides. So again, the spindle fibers are going to pull them apart, anaphase for apart. Same over here, we're going to have spindle fibers that are going to form, and they're going to basically condense and pull each sister chromatid away to the opposite ends of the cell. Okay, so we've split this chromosome now into 2 individual chromatids. Finally, for telophase 2, right, the nuclear envelope is going to reform over each chromatid. So the nucleus is going to reform around here. Cytokinesis is going to occur, and that cell is going to cleave and split apart into 4 new cells, each with 1 copy, 1 chromatid of each chromosome. So here was our original chromosome 1, here was our original chromosome 2, but we just have 1 chromatid. These are all haploid cells, right? So they only have 1 copy of each chromosome, so they're N, so they're haploid. Now, depending on if we're a male or a female, these will be sperm or egg cells. So this is kind of the generic version of what's happening. When we talk in the next 2 lecture videos, specifically looking at how sperm form and how oocytes form or eggs form, this process down here will differentiate just a little bit. But this is the overall process of meiosis, okay? We are starting with a single normal cell, a germ cell that's got 2 pairs of 23 chromosomes in a human, and we are dividing it into forming 4 cells. So even The thing that I think students get confused about is this is a division process because we're focusing on the genetic material. We're not focusing on the cells. So when we think about dividing, we think we would have less cells, but we actually are going to have more cells, but each one is going to have less genetic material in it. So again, we started with one cell, 2 copies of 23 chromosomes, so 46 chromosomes total. Each chromosome had 2 chromatids. We are then ending, we go through meiosis I, where we end up with 2 cells that have 1 copy of each chromosome. So in a human, there would be 1 copy of every one of the 23 chromosomes. Each of those chromosomes have 2 chromatids. And then meiosis II, where we're going to then break those chromatids apart into individual sister chromatids and make 4 cells, each with half the number of chromosomes, there's only 23, that can then be used in the process of fertilization. So make sure that you understand these different stages as you kind of read through those and read through the material that was presented, kind of going into detail some of these. But, you know, in general, the steps of prophase are in general the same things that are happening, whether it's mitosis, meiosis I, or meiosis II. The nuclear envelope starts to disintegrate. We get the spindle fibers that are going to start to form. Metaphase is generally the same process. You know, the chromosomes are going to line up along the middle. Now in meiosis I, the pairs are going to still be together, but chromosomes are going to line up along the middle. Anaphase, they're going to get separated to opposite poles. Telophase, the nucleus is going to reform, and then at some point the cell will cleave and split into 2, and then you're going to end up with daughter cells of some kind. So the general activity in these prophase, metaphase, anaphase, telophases are kind of the same depending on, you know, whether it's mitosis or meiosis I or meiosis II, but the end result is going to be very different because of basically what we're starting with. So in meiosis, we're making 4 cells, half the number of chromosomes that will be used in fertilization.